Good morning, students. Next class is purine and pyrimidine biosynthesis. The purine and pyrimidine are nucleic acid bases, and purine consists of two major classes, adenine and guanine, and pyrimidines consist of three major classes, cytosine, thymine, and uracil. You may be studied in your UG level, but this is the biosynthesis, how it is synthesized in microorganisms. So we have two different setup of the biosynthesis pathways. One is de novo pathway and salvage pathway. De novo pathway is nothing but synthesis of a complete the fragment of the complete fragment of the nucleic acid base. Okay, by each and every element. Okay, that is why it is called as de novo. De novo means starting from the scratch. Okay, that means from each and every element is used to frame, to coin, to build. Okay, the complete the nucleic acid base. Okay, that is why it is called as purine biosynthesis is de novo. This type is called as de novo synthesis, and second type is called as salvage. Already formed, already synthesized one is there, and that is used for another one. Okay, to make another type of nucleic acid base. So that is why it's called as purine salvage pathway. Salvage is already synthesized one is used to convert, okay, this one to another one, that is called as salvage. We can start from the first. Okay, that is the de novo synthesis. It is a very important question in the examination. No need to write all the structures, it's very complex, okay, for you. And PRPP, phosphoribosyl 5 pyrophosphate, and this is converted to phosphoribosyl and amino group, okay, phosphoribosyl amine, and by the action of glutamine to removal of 2 pyrophosphate, and a bunch of steps requiring glycine and CH2, tetrahydrofolate, and glutamine, ATP, carbon dioxide, aspartate, and formyl tetrahydrofolate. All the substances required to synthesize IMP, inosine monophosphate, and inosine monophosphate is converted into xanthine monophosphate, and xanthine monophosphate is converted to GMP, that is guanosine monophosphate, and guanine monophosphate is formed, okay, as a final, and this is the guanine 1. So this guanine is also involved in the conversion of guanine to GMP. Okay, this is the salvage pathway. You can see here. So till this formation of GMP, okay, that is purine biosynthesis de novo, and these three involved in the conversion of already available one, that is adenine to AMP, adenosine monophosphate, and adenine to adenosine, adenosine to inosine, and inosine to IMP. And second one is hypoxanthine to IMP, inosine monophosphate, and guanine to guanosine monophosphate. Okay, that is these three are the steps involved in the shortcut method. Okay, this already formed one, guanine is already there, means this will be converted into GMP. So hypoxanthine is already there, means that is converted into IMP by the incorporation of PRPP, that is phosphoribosyl pyrophosphate, and by the enzyme HGPRTase, and here the name of the enzyme APRTase. And the conversion of IMP to adenylosuccinate, adenylosuccinate to AMP, okay, by GTP and aspartate is required. Adenylosuccinate is synthesized, and removal of fumarate, this will lead to AMP. So this adenosine to AMP by ATP incorporation. Okay, so these are all the bunch of reactions, and till this set, okay, this is de novo till AMP and GMP formation, that is de novo synthesis, and salvage pathway starting from the bottom. Okay, and by expansion, okay, this is a salvage pathway, HGPRTase and ARPTase. HGPRTase is abbreviated as hypoxanthine guanine phosphoribosyl transferase, and it makes both IMP and GMP. Okay, and maybe this one is involved in the conversion of hypoxanthine or guanine, okay, with the phosphoribosyl pyrophosphate to IMP or GMP and pyrophosphate. So coming to the pyrimidine biosynthesis, the next base, that is cytosine, thymine, and uracil. So this cytosine, thymine, and uracil synthesized by de novo synthesis also. Okay, this one carbamyl phosphate, and the first compound is converted to carbamyl aspartate, and this enzyme is a very special enzyme, very unique enzyme, and a regulatory enzyme also, that is studied more extensively, okay, for the regulation of enzymes and the regulatory properties of enzyme, that is allosterism. Okay, allosterism means allo means two, that is two phases of the enzyme. Okay, one is substrate binding site and another one is regulator binding site. So this one enzyme is extensively studied by the scientists, and that is why we have the knowledge of allosterism. Carbamyl phosphate is converted to carbamyl aspartate, and this will be converted into sequence of biochemical reactions to orotate, and orotate to OMP, orotate monophosphate by PRPP, and orotate monophosphate to UMP, that is uridine monophosphate, and uridine monophosphate to uridine diphosphate, and uridine triphosphate, and uridine triphosphate to cytidine triphosphate, and deoxyuridine monophosphate, and deoxythymidine triphosphate. You can see here there are three different type of nucleic acid bases. One is cytosine, thymine, and uracil. You can see here the cytosine is formed here, and this is thymine and uracil. Okay, this is CTP, TTP, and UTP, all the end products of this one. And usually this TTP, that is thymine. So thymine is usually present in that is DNA in the deoxygenated form. Okay, that is why CTP is converted to UMP, that is deoxy-UMP, and deoxy-UMP is converted to deoxy-TTP. So the TTP is very, very important, because in the place of uracil, the thymine will be present. Okay, that is why that is very important. The thymine should be in the deoxygenated form. It is not in the TTP form. And UDP further converted into deoxy-UDP and deoxy-UMP and deoxy-TMP also. So either this way the TTP will be formed, or either this way, okay, TMP will be formed. Again, the TMP will be converted into TTP, thymine monophosphate, and converted to thymine diphosphate, and further triphosphate. And this step is very, very important, because deoxy-UMP to deoxy-TTP, so TMP. So this is by dihydrofolate reductase, and so this enzyme is very important to convert tetrahydrofolate. This is tetrahydrofolate and methyl tetrahydrofolate. So this is a methyl donor actually. So this methyl group donor is very, very important. Without this enzyme, for example, if the enzyme is not present, the methyl group is not transferred to deoxy-UMP, not converted to TMP. If the TMP is not converted, automatically there is no synthesis of DNA. So that is why the anti-metabolites are used, antibiotics are used to stop this one, okay, for the bacterial infections. So that means this will stop the synthesis of TMP. So next one is the pyrimidine salvage pathway. The uracil plus PRPP converts UMP and PPI, and only for the uracil, okay, this is. And next one is nucleoside phosphorylase, UCT, that is uracil, cytosine, thymine plus ribose 1-phosphate synthesizes nucleosides of respective bases plus PI. So with this we can conclude the nucleic acid bases, and next topic is cell wall biosynthesis.